Welcome to Science Store online classes.
Don't forget to like our video and subscribe our YouTube channel for all the updates.
Welcome to all my dear students. Today I am taking biology class, and in that we are studying cell, the unit of life. Okay, last class we have studied about the eukaryotic cell, structure of the eukaryotic cell, and also we have studied the cell membrane, how the membrane and what are the biochemical molecules which will make up the plasma membrane, and also we have studied the cell wall, what is the function as well as how the cell walls are formed, and that their functions are studied. And also we started with the endomembrane system. In endomembrane system, the group of organelles, they differ in their structure as well as their function, but their functions are coordinated. Okay, ಆ ಫಂಕ್ಷನ್ ಕೋಆರ್ಡಿನೇಷನ್ ಇರೋದ್ರಿಂದ ಆ ಆರ್ಗನಲ್ಸ್ ಅನ್ನೆಲ್ಲ ಒಂದು ಗ್ರೂಪ್ ಮಾಡ್ತಾರೆ. ದಟ್ ಗ್ರೂಪ್ ವಿಲ್ ಕಾಲ್ ಇಟ್ ಆಸ್ ಎಂಡೋಮೆಂಬ್ರೇನ್ ಸಿಸ್ಟಮ್. ಅಂಡರ್ ದಟ್ ವಿ ಹ್ಯಾವ್ ಸ್ಟಡಿಡ್ ದ ಎಂಡೋಪ್ಲಾಸ್ಮಿಕ್ ರೆಟಿಕ್ಯುಲಮ್, ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ದ ಎಂಡೋಪ್ಲಾಸ್ಮಿಕ್ ರೆಟಿಕ್ಯುಲಮ್, and we have studied the Golgi complex. Okay, we have studied the structure of Golgi complex, where it contains two main parts, two main types of the Golgi complex, that is cis phase, and two phases of the Golgi complex, that is cis phase and trans phase. Okay, and also usually these Golgi complex are sac like structure which is stacked, which is present one above the other. They are present parallel to one another, and also they are present near to the nuclear membrane. Okay, and here you can find two main parts. We have studied the size as well as two main phases of the Golgi complex. This phase, which is also called as forming phase, and also trans phase, which is a maturing phase. Here all these two phases are different, entirely different. Their functions as well as their structure entirely different, but they are interconnected. They are interconnected to one another. Okay, usually this Golgi, the main function of the Golgi complex is which act as a packaging material. So the main function of the Golgi complex is packaging of material. Whatever the important material present in the cell membrane or the important material synthesized by endoplasmic reticulum undergo packaging. That is called, and that later forms the Golgi complex. It may be targeted. These packaged materials or the packaged material can move from one place to intercellular movement. It may help for intracellular movement. It helps for intracellular movement of material, or it may be secreted out of the cells. Here these packaged materials or the materials required materials may move intracellularly, or it is also called as within the cell. ಸೆಲ್ಲಿನ ಒಳಗೆ ಒಂದು ಜಾಗದಿಂದ ಇನ್ನೊಂದು ಜಾಗಕ್ಕೆ ಹೋಗಿ ಸಪ್ಲೈ ಮಾಡಲಿಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡ್ತದೆ ಈ ಗಾಲ್ಗಿ ಕಾಂಪ್ಲೆಕ್ಸ್, ಅಂಡ್ ಆಲ್ಸೋ ಸೆಕ್ರೇಷನ್ಗೆ ಹೆಲ್ಪ್ ಮಾಡ್ತದೆ. ಯಾವುದೇ ಇಂಪಾರ್ಟೆಂಟ್ ಅಥವಾ ಅನ್ವಾಂಟೆಡ್ ಮೆಟೀರಿಯಲ್ಸ್ ಆರ್ ಎಂಜೈಮ್ಸ್, ಯಾವುದಾದ್ರೂ ಎಂಜೈಮ್ಸ್ ಅನ್ನು ಸೆಕ್ರೇಟ್ ಮಾಡಲಿಕ್ಕಿದ್ರೆ ದಟ್ Golgi complex will help for the secretion. Okay, this is intracellular targets or secretion outside the cell. The material to be packed is in the form of vesicles. Usually the material is to be packed is in the form of vesicle. Small vesicle type form ಆಗ್ತದೆ. So ಏನಾಗುತ್ತೆ, the materials which has to be packed, that forms a outer thin, it contains important materials, ಒಳಗೆ materials ಇರ್ತದೆ, and these are covered by thin membrane and form a vesicle like structure. it form a vesicle like structure. By this way this Golgi complex will pack up the materials. Okay, ಯಾವುದಾದ್ರು ಇಂಪಾರ್ಟೆಂಟ್, ಈಗ ಇಂಪಾರ್ಟೆಂಟ್ ಪ್ರೋಟೀನ್ ಅಥವಾ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ ಯಾವುದೋ ಇಂಪಾರ್ಟೆಂಟ್ ಫ್ಯಾಟ್ಸ್ ಸಿಂಥಸಿಸ್ ಆದರೆ ಏನಾಗುತ್ತೆ ಸೈಟೋಪ್ಲಾಸಮ್ ಅಲ್ಲಿ ಇದ್ರೆ, ಸೊ ಇಟ್ ಈಸ್ ಈಸಿಲಿ ವೇರಿಯಸ್ ಎನ್ಸೈಮ್ಸ್ ಇರ್ತದೆ, ಈಸಿಲಿ ಇಟ್ ವಿಲ್ ಡೈಜೆಸ್ಟ್. So in order to prevent that, the particular type of material should be packaged or it should be separated from rest of the organelles or rest of the cytoplasmic content. ಆ cytoplasmic content ಇಂದ separate ಆಗಬೇಕು, ಹಾಗಾಗಿ ಅದನ್ನ vesicle ಆಗಿ ಫಾರ್ಮ್ ಮಾಡ್ತದೆ. So this is material vesicle and usually these vesicle, the packaged or the important materials are supplied from endoplasmic reticulum. Usually the important function of the endoplasmic reticulum is protein synthesis as well as fat synthesis. Alva, in class we have studied the important function of endoplasmic reticulum is protein synthesis because it contain ribosomes alva, ribosomes iರ್ತದೆ and also it is very near to the nuclear membrane ಗೆ ಹತ್ತಿರ ಇರೋದ್ರಿಂದ it is active, they are active in protein synthesis. So the synthesized protein is stored in the form of or packed in the form of vesicles. Later this vesicle will attach us to the cisternae of the Golgi complex ಇದು cisternae ಇರ್ತದಲ್ಲ, this sac-like structure or disc-like structure, we'll call it as cisternae, that will fuses with the cisternae. Okay, and move to the maturing phase. Here it will fuses with the cisternae. So illi bandu it will fuses with the cisternae. Usually ಈ cisternae concave shape ಅಲ್ಲಿ ಇರ್ತದೆ, forming phase ಅಲ್ಲಿ ಇರ್ತದೆ, or it is in cis phase. They are usually in cis phase. Okay, they are usually in cis phase. Okay, the material which is directly from the endoplasmic, that's why you can see the Golgi complex near the nuclear membrane. I have drawn the structure of the cell alva, cell structure draw ಮಾಡುವಾಗ ಹೆಚ್ಚಾಗಿ Golgi complex nucleus ಹತ್ರ develop ಆಗಿರ್ತದೆ. They are arranged near the nuclear membrane, that's why the protein, synthesized protein or endoplasmic reticulum ಹತ್ರ ಇರುವ ಕಾರಣ ಅದು ಸಿಂಥಸೈಸ್ ಮಾಡಿದ ಪ್ರೋಟೀನ್ಸ್ ಅನ್ನ cis phase ಅಲ್ಲಿ ಇರುವ ಸಿಸ್ಟರ್ನಿಗೆ ಬಂದು ಬರ್ತದೆ and it will attaches and releases the proteinaceous or the synthesized material inside the cisternae. Okay, before secreting they undergo sequential modification. Here the protein or the materials undergo modification inside the cisternae and later it form a maturing phase. Okay, so undergo modification inside the cisternae and it will convert into trans phase. It will move into trans phase of the cisternae. Here they undergo a trans phase and after the modification will be complete in the trans phase. It is concave shape. Here in trans phase the Golgi complex structure will be in a concave shape. Illi convex shape ಅಲ್ಲಿ ಇರ್ತದೆ. So modification ಆಗ್ತಾ ಹೋದಾಗೆ trans phase ಗೆ ಅದು ಹೋಗ್ತದೆ. Trans phase ಅಲ್ಲಿ ಏನಾಗ್ತದೆ, it will form a concave, it assumes a concave shape and it contains a modified food materials or modified proteins, modified fats will be present in trans phase. So in this phase you can see that particular protein, modified protein will release out of the Golgi complex. ಎಲ್ಲಾದರೂ ರಿಕ್ವೈರ್ಮೆಂಟ್ ಇದ್ರೆ ಈ modify ಆದಂತಹ proteins ಅಥವಾ lipids ಯಾವುದೇ materials ರಿಲೀಸ್ ಆಗ್ತದೆ ಸೆಲ್ಲಿಗೆ. So they start to perform various function. ಅಂದ್ರೆ ಇಲ್ಲಿ usually proteins, all the organelles, all the biochemical structures, enzymes, all these organelles are made up of proteins. Okay, the function, the mediation, the life process is taking place by the help of proteins, mainly by the help of protein. Protein ಎಲ್ಲದಕ್ಕೂ ನಮ್ಮ ಬಾಡಿಯಲ್ಲಿ ಯಾವುದೇ ರಿಯಾಕ್ಷನ್ ಅನ್ನ ರೆಪ್ರೆಸೆಂಟ್ ಮಾಡಲಿಕ್ಕೂ ಕೆಮಿಕಲ್ ರಿಯಾಕ್ಷನ್ ಆಗ್ತದೆ. ಆ ಕೆಮಿಕಲ್ ರಿಯಾಕ್ಷನ್ಸ್ಗೆ ಪ್ರೋಟೀನ್ is very important component. So here these proteins present inside the transphase will released when there is a requirement of the cell requirement ಗೆ ಬೇಸಿಸ್ ಅಲ್ಲಿ ಇದು ರಿಲೀಸ್ ಆಗ್ತದೆ. ಸೊ ಇಟ್ ಮೇ ಬಿ ಕನ್ವರ್ಟ್ ಇಂಟು ಪ್ರೋಟೀನ್ಸ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗ್ಬೋದು, ಬೇರೆ ಯಾವುದಾದ್ರೂ ಕಾಂಪೊನೆಂಟ್ಸ್ ಸೆಕೆಂಡರಿ ದ ಸೆಲ್ ವಾಲ್ ಕಾಂಪೊನೆಂಟ್ಸ್ ಆಗಿ, ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ಆಗಿ, ಅಥವಾ ಬೇರೆ ಬೇರೆ ಕೆಮಿಕಲ್ ರಿಯಾಕ್ಷನಿಗಾಗಿ ಪ್ರೋಟೀನ್ ಇಲ್ಲಿ ರಿಲೀಸ್ ಆಗ್ತದೆ. ಸೊ ದ್ಯಾಟ್ ಈಸ್ ಲೇಟರ್ ಯೂಸ್ಡ್ ಫಾರ್ ವೇರಿಯಸ್ ಬಯೋಕೆಮಿಕಲ್ ರಿಯಾಕ್ಷನ್ಸ್. ಓಕೆ, so that's why you can see the Golgi complex near the nuclear membrane. And next we'll move to this Golgi complex is also important site of synthesis of glycoproteins and glycolipids. Glyco is nothing but sugar, lipid plus lipids. Sugar plus lipid component is collectively called as glycolipids. Sugar plus proteins called as glycoproteins. Here the main function of the Golgi complex is synthesis of glycoproteins and glycolipids. Glycolipids matte glycoproteins anna synthesis ಮಾಡ್ತದೆ. Okay, these are the important function of the Golgi complex. ಯಾವುದು ಎರಡು phases importance is phase entrance phase. Here the vesicle, the important materials synthesized in endoplasmic reticulum is in the form of vesicle. They are collected inside the vesicle. This vesicle attaches to the cis phase of the Golgi complex or cis phase of the cisternae, and they undergo modification. So modification inside the cis phase alli it undergo formation modification, so that will convert into trans phase. Okay, and here in trans phase you can see the modified proteins as well as lipids. Later they get released from the Golgi complex when there is a requirement of the cell requirement iruvaga illiruvanತಹ content anna Golgi complex release ಮಾಡ್ತದೆ. So this is a main function of the Golgi complex. Next we'll move to the one more important organelle, endomembrane system, that is vacuole. The one more important system, that is lysosome. Before that we'll see lysosomes. Okay, these lysosomes are the membrane bound vesicular structure. These lysosomes are nothing but the membrane bound vesicular structure. Membrane, it is outerly covered by membrane bound vesicular structure formed by the process of packaging in the Golgi complex. Golgi complex alli enagutte packaging. The main function of the Golgi complex is packaging of material. So here mainly these lysosomes are formed by the packaging of materials by Golgi complex. So what happens inside this, if you the detailed study the isolated lysosomes have found to be containing many type of hydrolytic enzyme. Here these lysosomes are nothing but vesicular structure, which is these lysosomes are very rich on the keen observation or on the detailed study reveal that these lysosomes contain large number of hydrolytic enzymes. So hydrolytic enzymes are nothing but which mediate the hydrolysis of the chemical reaction. Hydrolysis andre chemical components anna hydrolysis madudu. The chemical bond is broken. Hydrolysis means the chemical bond will be broken by the addition of water to the compounds. Enagutte illi chemical compounds idre 2 A ಮತ್ತೆ A plus B Here you can find A, B chemical components. It is two different types of chemical components or chemical compound. It is a chemical compound. By the addition of water, what happens? This will broken down into, this chemical compound will broke down into, they are bond. The bond present in between these will breaks, so it form two different components. Okay, this type of reaction where is called as the breaking of bond by the addition of water is called as hydrolysis. So this, the main function of hydrolytic enzymes are hydrolysis. It is also called as hydrolysis. This is also, hydrolytic enzymes are also called as hydrolases, which contain different types of hydrolytic enzyme inside the lysosome. Lysosome ಒಳಗೆ ಬೇರೆ ಬೇರೆ ತರದ hydrolytic enzymes ಇರುತ್ತೆ. Few of them are proteases, lipases, carbohydrases. Okay, carbohydrates, protease, lipase. Okay, lipase, carbohydrates. These are the different types of hydrolases or hydrolytic enzyme, and they are active at optimal acidic pH. Only active at they are active at optimal acidic pH. They are active only at the optimal acidic pH. Optimally active at acidic pH. Usually acidic pH alli active ಆಗಿರ್ತದೆ. Because all the time enzymes active ಇದ್ರೆ ಏನಾಗುತ್ತೆ? The reaction continuously takes place. So these enzymes usually active only at the acidic pH. pH acidic ಆಗಿರುವಾಗ ಮಾತ್ರ ಆಕ್ಟ್ ಆಗುತ್ತೆ. Normal pH ಅಲ್ಲಿ ಆಕ್ಟ್ ಮಾಡಿದ್ರೆ ಯಾವಾಗಲೂ chemical reaction ಆಗ್ತಿರುತ್ತೆ. That may be lead to the that is not required for the cell ಅಲ್ವಾ. ಸೆಲ್ಲಿಗೆ ಅದು ಅಗತ್ಯ ಇರುವುದಿಲ್ಲ. ಕಂಟಿನ್ಯೂಸ್ ರಿಯಾಕ್ಷನ್ ಆದರೆ ನಾರ್ಮಲ್ ಫಂಕ್ಷನಿಂಗ್ ಪ್ರಾಬ್ಲಮ್ ಆಗ್ತದೆ. ಸೆಲ್ಲಿದೊಳಗೆ ದ ಫಂಕ್ಷನ್ ವಿಲ್ disrupted, the normal functioning of the cell disrupted. So in order to prevent that, these enzymes are only active at the acidic pH. The main function of hydrolases is proteases will break down the proteins, lipases will break down the lipid molecules ಅನ್ನು ಬ್ರೇಕ್ ಮಾಡ್ತದೆ. Carbohydrases will break the carbohydrate ಅನ್ನ ಬ್ರೇಕ್ ಮಾಡ್ತದೆ, carbohydrases. And also nucleic acid is also broken down by nucleases. So these main function of hydrolytic enzymes are breaking down of proteins, lipids, carbohydrate, nucleic acids, okay, etc. You can see these are the complex biomolecules alva, complex chemical compounds, protein, lipids ಎಲ್ಲ long chain ತುಂಬಾ chemicals ಎಲ್ಲ ಸೇರಿ ಒಂದು components ಆಗ್ತದೆ. So ಇದನ್ನ ಬ್ರೇಕ್ ಮಾಡಿದಾಗ ಏನು ಸಿಗ್ತದೆ? ಎನರ್ಜಿ ಸಿಗ್ತದೆ. So and also for various metabolic activities ಗೆ ಈ biomolecules ಎಲ್ಲ important. So ಇದನ್ನ ಬ್ರೇಕ್ ಮಾಡಲಿಕ್ಕೆ enzymes ಬೇಕು. ಆ enzymes ಎಲ್ಲಿ ಇರ್ತದೆ? Lysosomes ಒಳಗೆ ಇರ್ತದೆ. So this is the main function: proteins, lipids and nucleic acids. Okay, capable of breaking down of protein, lipids and carbohydrate. Nucleic acid is broken down by nucleases. Okay, nucleases nucleic acid ಅನ್ನ ಬ್ರೇಕ್ ಮಾಡ್ತದೆ. So these are the different types of hydrolases or hydrolytic enzyme. ಇದೆಲ್ಲ ಯಾವುದು? ಬೇರೆ ಬೇರೆ ತರದ hydrolytic enzymes. ಅದು ಯಾವಾಗ ಆಕ್ಟಿವ್ ಆಗಿರ್ತದೆ? Only in acidic pH. This is about the lysosome. Next we'll move to the one more important organelle, that is vacuole. One more important organelles is vacuoles. Okay, vacuoles, here it is also a type of membrane bound organelles. Okay, it is also membrane bound organelles, bound space. Here the vacuole, membrane bound space inside the cytoplasm. It is nothing but the membrane bound space inside the cytoplasm. Here in plant cells, maximum area of the cytoplasm is covered by this vacuole. The large amount of large area of the cell is covered with the occupied with the this. If you consider this is a plant cell, okay, large area is occupied by the vacuole. Usually these vacuoles contain water, sap is nothing but the dissolved minerals as well as salts. Okay, sap enta minerals, dissolved minerals, salts, food materials, collective will generally call it as sap. Okay, vascular system alli sap conduction anta heltevalla. Xylem will conduct the sap which is contain dissolved minerals. Dissolved mineral salts anna ನಾವು ಏನು ಜನರಲ್ ಆಗಿ sap ಅಂತ ಹೇಳ್ತೇವೆ. Water and excretory materials. Usually these or excretory or waste material. Usually vacuoles contain water, the large amount of water, sap, and also contains the excretory materials which is not required for the cell, can be stored in the inside the vacuoles. The main function of this vacuole, here these vacuoles are outerly covered by a thin plasma membrane. Here all these organelles are covered by a membrane which is nothing but a plasma membrane. Lipid bilayer in the form ಆಗಿರ್ತದಲ್ಲ ಅದೇ membrane ಎಲ್ಲ organelles ಸುತ್ತ ಇರುವಂತದ್ದು. So this outer thin layer of the vacuole is called as stonoplast. Very important. The outer thin membrane of the vacuole is called as stonoplast. Okay, it is also called as stonoplast. In plants, vacuoles occupy 90% of the, in plant cell 90% of the cytoplasmic space is occupied by vacuole. So you can see the you can also see the nucleus in a peripheral region. Animal cell elli irtade, central irtade. Because of the presence of vacuole in plant cell, the nucleus is pushed to the periphery of the cell. ಅಲ್ವಾ, ಈ ಜಾಸ್ತಿ ಏರಿಯಾ ವ್ಯಾಕ್ಯೂಲ್ ಕವರ್ ಮಾಡೋದ್ರಿಂದ ಏನಾಗ್ತದೆ, ಈ ನ್ಯೂಕ್ಲಿಯಸ್ ಇರ್ತದಲ್ಲ ಸೆಂಟ್ರಲ್ ಇರಬೇಕಾದ ನ್ಯೂಕ್ಲಿಯಸ್ ಪೆರಿಫರಿಗೆ ಪುಷ್ ಮಾಡಿರ್ತದೆ. ಸೊ ಬಿಕಾಸ್ ಹಿಯರ್ 90% ಆಫ್ ದ ಸೆಲ್ ಇಸ್ ಕವರ್ಡ್ ವಿತ್ vacuole. Okay, so that play very important role in plant cell. Okay, 90% of the total volume of the cell. In here in plants, the main function of the vacuole here, the tonoplast facilitate the transport of number of ions and other material. Here this tonoplast ideyalla, outer vacuole is the outer membrane, it will facilitate the movement or transport of the ions and other materials, ions and other components in and out of the cell. Okay, here it will facilitate the movement of the ions or components in and out of the cell against the concentration gradient into the vacuole. Here the usually that in turn that will help the cytoplasm. Enaagtade, the ions and other components here move. Tonoplast will facilitate their movement against the concentration gradient. I have mentioned concentration gradient andre higher matte lower concentration. Two concentration ide, higher, this is higher concentration, this is lower concentration. Okay, this is separated by a membrane system. This will generally call it as concentration gradient. Variation in the concentration, adanna ನಾವು concentration gradient ಅಂತ ಹೇಳ್ತೇವೆ. Concentration ಅಲ್ಲಿ variation ಇದೆ, difference ಇದೆ, ಅದನ್ನ ನಾವು concentration gradient ಅಂತ ಹೇಳ್ತೇವೆ. Here the movement of ions and other components is against the concentration gradient, that is move inside the vacuole. Usually ಇಲ್ಲಿ ಏನಾಗ್ತದೆ, concentration ಗೆ ವಿರುದ್ಧವಾಗಿ higher, ಇಲ್ಲಿ ಒಂದು here you can see the higher concentration, the more ions or the components will move inside the inside the vacuole. So that is facilitated by Stonoplast. Stonoplast ಏನು ಮಾಡ್ತದೆ, ಕಾನ್ಸಂಟ್ರೇಷನ್ ಗ್ರೇಡಿಯಂಟ್ ಅಗೇನ್ಸ್ಟ್ ಆಗಿ ಅಯಾನ್ಸ್ ಅನ್ನು ಮೂವ್ ಮಾಡಲಿಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡ್ತದೆ, ಫೆಸಿಲಿಟೇಟ್ ಮಾಡ್ತದೆ, ಹೆಲ್ಪ್ ಮಾಡ್ತದೆ. ಸೊ that will maintain the cellular structure, cellular function ಅನ್ನು maintain. By this they will maintain the cellular function. ಅದರಿಂದಾಗಿ cellular function ನಾರ್ಮಲ್ ಆಗಿ ನಡೀಲಿಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡ್ತದೆ. ಆ್ಯಂಡ್ ಕಂಪೇರ್ಡ್ ಟು ಔಟರ್ ಸೈಟೋಪ್ಲಾಸಮ್, ಯೂಶುವಲಿ ಇನ್ಸೈಡ್ ದ ವ್ಯಾಕ್ಯೂಲ್ ಯು ಕ್ಯಾನ್ ಸಿ ಹೈ ಕಾನ್ಸಂಟ್ರೇಷನ್. ಯಾವುದೇ ಅಯಾನ್ಸ್ ಅಥವಾ ಮಿನರಲ್ಸ್ ಯಾವುದೇ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಕಂಪೇರ್ ಮಾಡಿದರೆ ಈ ವ್ಯಾಕ್ಯೂಲ್ ಒಳಗೆ ಜಾಸ್ತಿ ಇರ್ತದೆ, ಬಿಕಾಸ್ ದ ಮೂಮೆಂಟ್ ಆಫ್ ದ ಅಯಾನ್ಸ್ ಅಗೇನ್ಸ್ಟ್ ದ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಗ್ರೇಡಿಯಂಟ್ ಅಲ್ವಾ. ಯೂಶುವಲಿ ಏನಾಗುತ್ತೆ, ಹೈಯರ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಇಂದ ಲೋವರ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ಗೆ ಹೋಗ್ತದೆ ಅಲ್ವಾ. ಅಯಾನ್ಸ್ ಅಥವಾ ಮಾಲಿಕ್ಯೂಲ್ಸ್, ವಾಟರ್ ಏನೇ ಆಗಲಿ, ಎರಡು ಬೀಕರಲ್ಲಿ ವಾಟರ್ ಇಟ್ಟರೆ, so here consider two beakers which is separated by a thin membrane ಇದೆ. So here you fill it up with water. ಸೊ ಏನಾಗುತ್ತೆ, ಇಲ್ಲಿ ವಾಟರ್ ಇಲ್ಲ ಆದರೆ ಏನಾಗುತ್ತೆ, ತಿನ್ ಮೆಂಬ್ರೇನ್ ಅಥವಾ ಒಂದು ಕ್ಲಾತಲ್ಲಿ ಸೆಪರೇಟ್ ಮಾಡಿ ಏನಾಗುತ್ತೆ, ಇಲ್ಲಿಂದ ವಾಟರ್ ಎಲ್ಲ ಇಲ್ಲಿಗೆ ಮೂವ್ ಆಗುತ್ತೆ, ಹೈಯರ್ ಕಾನ್ಸಂಟ್ರೇಷನಿಂದ ಲೋವರ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಆಗಿ ಮೂವ್ ಆಗ್ತದೆ. ಸೇಮ್ ವೇ ದ ಅಯಾನ್ಸ್ ಆರ್ ದ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ವಿಲ್ ಮೂವ್ ಹೈಯರ್ ಕಾನ್ಸಂಟ್ರೇಷನಿಂದ ಲೋವರ್ ಕಾನ್ಸಂಟ್ರೇಷನಿಗೆ ಹೋಗುತ್ತೆ. ಬಟ್ ಹಿಯರ್ ಏನಾಗುತ್ತೆ, ಅಯಾನ್ಸ್ ಲೋವರ್ ಕಾನ್ಸಂಟ್ರೇಷನಿಂದ ಹೈಯರ್ ಕಾನ್ಸಂಟ್ರೇಷನಿಗೆ ಬರ್ತದೆ. ದಟ್ ಈಸ್ ಅಗೇನ್ಸ್ಟ್ ದ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಗ್ರೇಡಿಯಂಟ್. ಅದರಿಂದಾಗಿ ಏನು ಮಾಡ್ತದೆ, ಇದು ಸೆಲ್ಯುಲಾರ್ ಫಂಕ್ಷನ್ ಅನ್ನು ಫೆಸಿಲಿಟೇಟ್ ಮಾಡ್ತದೆ. ಹಾಗಾಗಿ ವ್ಯಾಕ್ಯೂಲ್ ಅಲ್ಲಿ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಜಾಸ್ತಿ ಇರ್ತದೆ. ದೇರ್ ಇಸ್ ಅ ಹೈ ಕಾನ್ಸಂಟ್ರೇಷನ್ ions inside the vacuole. Usually the concentration is very high inside the vacuole. ವ್ಯಾಕ್ಯೂಲ್ಗೆ ಕಂಪೇರ್ ಮಾಡಿದ್ರೆ ಏನಾಗುತ್ತೆ, ದ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಈಸ್ ವೆರಿ ಹೈ. ಯು ಕ್ಯಾನ್ ಸಿ ಹೈ ಕಾನ್ಸಂಟ್ರೇಷನ್. ಓಕೆ, ಇನ್ ಸಮ್ ಅದರ್ ಲೋವರ್ ಆರ್ಗ್ಯಾನಿಸಮ್, ಓಕೆ, ಸಿ ಅದರ್ ಟೈಪ್ ಆಫ್ ವ್ಯಾಕ್ಯೂಲ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಕಾಂಟ್ರಾಕ್ಟೈಲ್ vacuole. Contractile vacuole usually present in protista, mainly amoebadalli ಈ contractile vacuole ಇರ್ತದೆ. So this contractile vacuole present inside the amoebal cell, amoeba cell ಒಳಗೆ contractile vacuole ಇರ್ತದೆ. So that mainly help for the osmoregulation, that helps for this contractile vacuole. The main function is osmoregulation. ಸೊ ಯೂಶುವಲಿ ಈ ಅಮೀಬಾ ಎಲ್ಲ ಏನು ಮಾಡ್ತದೆ, ವಾಟರ್ ಅಕ್ವಾಯಸ್ ಮೀಡಿಯಮ್, ಅಕ್ವಾಟಿಕ್ ಮೀಡಿಯಮ್ ಅಲ್ಲಿ ಗ್ರೋ ಆಗ್ತದೆ, ವೇರ್ ದೇರ್ ಇಸ್ ಅ ಹೈ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಆಫ್ ವಾಟರ್. ಸೊ ವಾಟ್ ಹ್ಯಾಪನ್ಸ್, ದ ಎನಿ ಅಯಾನ್ಸ್ ಆರ್ ದ ರಿಕ್ವೈರ್ಡ್ ಮೆಟೀರಿಯಲ್ಸ್, ಇಫ್ ಇಟ್ ಈಸ್ ಕನ್ಸಿಡರ್ ದಿಸ್ ಇಸ್ ಅ ಅಮೀಬಲ್ ಸೆಲ್, ಕ್ಯಾನ್ ಸಿ ದ ಕಾಂಟ್ರಾಕ್ಟೈಲ್ ವ್ಯಾಕ್ಯೂಲ್ ಲೈಕ್ ದಿಸ್. ದಿಸ್ ಇಸ್ ಅ ನ್ಯೂಕ್ಲಿಯಸ್ ಹೀಗಿರ್ತದೆ. ಸೊ ನಿಯರ್ ದ ನ್ಯೂಕ್ಲಿಯಸ್ ಆರ್ ಇನ್ಸೈಡ್ ದ cytoplasm you can see the contractile vacuole. What happens when there is a requirement of when there is a removal of waste material, excess ions will move to the contractile vacuole. This will move near the membrane and releases out the materials or the excess, secrete the materials, unwanted material will be secreted. So ಏನಾಗುತ್ತೆ, ಇಲ್ಲಿ ಅಯಾನ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಕಮ್ಮಿ ಆದ ಕೂಡಲೇ ಸೈಟೋಪ್ಲಾಸಮ್ ಅಲ್ಲಿ ವಾಟರ್ ಕ್ಯಾನ್ ಈಸಿಲಿ ಎಂಟರ್. ಸೊ ಎಂಟರ್ ಆಗುತ್ತೆ ಈಸಿಲಿ. when there is no requirement of water, this will release the ions. ಹಾಗೆ ಏನಾಗುತ್ತೆ, ಮೇನ್ಲಿ ಒಳಗೆ ಆಸ್ಮೋರೆಗ್ಯುಲೇಷನ್ ಅಂದರೆ ವಾಟರ್ ರೆಗ್ಯುಲೇಷನ್, ಆಸ್ಮೋಟಿಕ್ ಪ್ರೆಷರ್ ಅಂತ ಬರ್ತದೆ. ಆಸ್ಮೋಸಿಸ್ ಅಂದರೆ ದ ಮೂಮೆಂಟ್ ಆಫ್ ವಾಟರ್. ಹಿಯರ್ ದಿಸ್ ಕಾಂಟ್ರಾಕ್ಟೈಲ್ ವ್ಯಾಕ್ಯೂಲ್ ವಿಲ್ ಫೆಸಿಲಿಟೇಟ್ ದ ಮೂಮೆಂಟ್, ದಟ್ ರೆಗ್ಯುಲೇಟ್ ದ ಮೂಮೆಂಟ್ ಆಫ್ ವಾಟರ್ ಇನ್ ಆ್ಯಂಡ್ ಔಟ್ ಆಫ್ ದ ಸೆಲ್ ಬೈ accepting by or taking up the ions present in the cytoplasm. cytoplasm ಅಲ್ಲಿ ಇರುವಂತಹ ಅಯಾನ್ ಅನ್ನು ತಕೊಂಡು ಆಸ್ಮೋರೆಗ್ಯುಲೇಷನ್ಗೆ ಹೆಲ್ಪ್ ಮಾಡ್ತದೆ. ಓಕೆ, ಆಸ್ಮೋರೆಗ್ಯುಲೇಷನ್ಗೆ ಹೆಲ್ಪ್ ಮಾಡ್ತದೆ. and also one more in protista, in some protistans you can see the presence of food vacuoles. Can also see the presence of food vacuoles which engulf usually the membrane will engulf the food material, okay, food material, and form the small vesicle like structure, and this vesicle like structure we'll call it as food vacuoles. Inside that vacuole you can see the presence of food material, okay, formed by engulfing the food material. Food ಅನ್ನು engulf ಮಾಡ್ತದೆ, ಅದು separate ಒಂದು ವೆಸಿಕಲ್ ಅಲ್ಲಿ ಇರ್ತದೆ. So that is called as food vacuole. That we'll call it as food vacuole. Okay.
Don't forget to like our video and subscribe our YouTube channel for all the updates.